Hello, everyone, and welcome to another episode of Psychedelic Insights. Today, I have the pleasure of conducting an interview with Jim Gilligan, the CEO of Tryp Therapeutics. As a leader in the industry, Jim brings a wealth of knowledge and expertise to the table. We'll be exploring the groundbreaking work being done at Tryp Therapeutics and gaining valuable insights into their initiatives and projects. Please join us for what promises to be a highly informative and educational discussion.
Thanks, Adam. I appreciate the opportunity to speak with you.
All right, awesome. So to get things started, let's just kick things right off. What differentiates Tryp Therapeutics from other companies in the psychedelic space, and what gives you the confidence that Tryp will ultimately be successful? What are the near-term catalysts from Tryp that investors should be looking forward to?
All good questions. So I'll start from the top. One of the things that we decided from the beginning, and given my background in the biotech industry and the pharma industry and our others, is that our goal is to develop psychedelics as ethical pharmaceuticals in conjunction with psychotherapy. So we're looking at performing clinical studies and generating data that supports their use in different indications. So we're going down that particular route. The other thing, we did start a little bit later than some of the other companies. So we decided, what if we look at other areas of unmet medical need that where psychedelic assisted psychotherapy could have a benefit? And we spoke with a number of folks in the field. We'll talk about a little bit later. Robin Carhart Harris is head of our SAB, and really spend some time trying to think, where might this approach, this new treatment paradigm, have a benefit, and especially in areas where there aren't many options, many opportunities for patients. So the two major areas we picked were eating disorders, and the first being binge eating disorder. And of course, there could be other opportunities in the obesity space. And the next was chronic pain, but in particular nosoplastic pain. And we'll get to that a little bit later as well. But just to start off with, nosoplastic pain is pain that originates in the brain. And the best example of that is phantom limb, where through a terrible trauma or tragedy, someone loses an extremity. It could be in a car accident, it could be a service person. And yet later on, they have really persistent pain in an arm or leg that no longer exists. So certainly the signal, the sense of pain has to originate in the brain. That's one of our targets. And most recently, we mentioned that we have a new collaboration with Mass General Hospital and Harvard on irritable bowel syndrome. The focus there is on abdominal tenderness or abdominal pain, visceral tenderness, which again fits into that pain bucket. So those are our two main areas of research that we feel right now we're leaders in. We have first mover advantage. We were the first to go and identify those areas as potential indications or targets. And, we want to examine the utility. There is a certain risk there, and we'll get to that. But, we went down a path less trodden, but there's certain risks. Will it work there? And we can discuss why we think it will work there as part of the discussion.
Thanks. That's really interesting. I actually have a friend of mine. He had his arm amputated, and he was experiencing tremendous amounts of phantom limb pain, which was really, really debilitating for him. And after just one breakthrough psilocybin session, he reports that he never, ever had pain in where that limb was again, which is amazing.
It is. So my roommate from college, orthopedic surgeon, and it was one of the first times he explained that to me, that, certain times people have amputations. And then you can imagine, as a surgeon, the patient comes back and says, "Hey, doc, I have a problem. That leg you cut off, I have pain in that leg." And we, one of our scientific advisors is Joe Castellanos at UCSD. He has a particular interest in phantom limb. And there's a lot of anecdotal information in that particular indication. And certainly, think about how outstanding that would be to alleviate these people's concerns with this chronic pain after having gone through a trauma like that. So that's certainly something we're interested in.
Yeah, it's life-changing.
Yeah.
Next question. So the well-known Robert Carhart Harris is the chairman of Tryp's Scientific Advisory Board. Can you please talk about Tryp's relationship with Robin and the value of his contributions to the company so far?
So I can talk from a personal perspective. When I first came to Tryp, they had an oncology asset that I had helped them with, and then they asked for me to look at their PFN program, psilocybin for neuropsychiatric disorders, and I freely admitted that I haven't really worked that much in the CNS sector. I've worked cardiovascular, endocrine, different. And they said, well, take a look at it. So I had to do a lot of reading because that wasn't my area of expertise. And of course, one of the articles you're always reading about are from Robin Carhart Harris. And it just so happened they had already brought him on as the head of the Scientific Advisory Board. And Robin was incredibly generous with his time. I was literally speaking to him every other week when he was still in London, just having him explain to me his vision of the value of psychedelics in treatment and how he felt that the field would advance, and also the ideas we had with respect to nosoplastic pain, with respect to eating disorders. And he's just been a tremendous resource. And I mean, I just had dinner with him out at JP Morgan a couple of weeks ago. And, we're always, we just value our banter back and forth and talking about the advances in the whole field. And certainly he's been instrumental when we were developing 8803 to come up with the attributes of the drug that he felt would be beneficial in advancing treatment. And he's just a very valued resource when we need to do things. We're giving another example. We do a lot of neuroimaging in our studies, trying to understand what happens in the brain. And so we're pulling together different folks we work with to just have a sit down, brainstorming to go through the neuroimaging, and Robin will be a part of that. So he's a constant resource for us. And, he just brings so much to the table. We're very, very lucky to have him as part of our team.
That's awesome. And yeah, I think that he's extremely well respected within the psychedelic communities in general. And he's a huge resource in terms of the knowledge that he has. In terms of, can you give any insight in terms of when you guys first kind of told him about IV psilocybin, what his reaction was like?
So it was funny. So I'll take a step back. One of the things that, for me, I've been in this field for over 35 years. And what you want to do, really, you want to reduce risk, right? You want to try to ensure that you have the best possible chance of success in the clinic. And when one of my discussions with Robin, he relayed that one of his issues is, in certain circumstances, he would like to be able to give maybe a little bit higher dose or get a little bit higher exposure, because I'll never forget this. He explained the subconscious as maybe a series of doors that you need to go through different levels in order to get to what could be causing the problem. And that due to the variable nature of the blood levels, he really couldn't dose escalate reliably. And that just piqued my curiosity. I have a PhD in pharmacology, so I wanted to go back and look at what could be causing that, what could attribute that. And the anecdote he told me is, he said, Jim, I could be in a situation where there's someone who weighs maybe 60 kilos, give them the capsule, and after an hour or so, they're looking at me like, is this all that happens? And then someone your size comes in and they have a full-blown experience. So in looking at it, the first thing is that psilocybin, which is in mushrooms, is not the active molecule that goes to the brain, and your body gets converted to psilocin. So you could have variability there, certainly, that could contribute to differences in blood levels. And then the other contributing factor is anything that you swallow and goes into your stomach intestines. The blood that leads from the GI tract goes into the liver, and you get what's called first pass metabolism. And so you can have variability there. and there's a number of other factors that could contribute to it. But what really cemented it for me was there's a publication called Brown et al. And they try to do things very accurately. They weighed the people and they dosed them by weight. So, for example, if you weighed 50 kilos and you got a 0.3 mg per kg dose, that's a 15 mg dose. If, on the other hand, you weighed 100 kilos, you would get a 30 mg dose. And so they did that for different ranges. So 0.3 mg per kg, 0.45 mg per kg, and 0.6 mg per kg. And the data, Dr. Paul Hudson, University of Wisconsin, had the data, and he shared it with me. And one of the first things I noticed was that there were some people who received the 0.3 mg per kg dose, had higher blood levels than people who got the 0.6 mg per kg dose. So certainly there's variability in blood levels. And, I gave you an example of a couple of things that can contribute. Obviously, there's others, but how do you address that? How do you make certain that the patient is getting the requisite blood levels of psilocin, which is what goes into the brain? And normally, you would never go from something that's convenient, like oral, to an IV. But if you ask anybody in medical research, the most accurate way to dose is obviously IV. And if you want to give the most accurate dose, you would give psilocin, not the precursor, because that's the actual molecule. So what had happened was I was just going back and forth with Robin, and I hadn't shared with him yet what I was thinking about. And so we're having a call, and he goes, Jim, I think I figured it out. I think what you're going to do is you're going to give an IV infusion of psilocybin. And I go, close. He said, close? I go, yeah, I'm going to give IV psilocin. And he just said, oh, that's brilliant. That's great, because that's even more accurate. The other reason we started looking at it is when again, Robin and other people in the field, they support the fact that you need to have the psychedelic experience in order to derive a full clinical benefit. And I started thinking about, what if someone's having a really profound experience and the drug runs out? You can't put a quarter in the machine. It is what it is. What if you could just continue the infusion so that they could complete their journey, they can complete their experience? On the other side, if there's a problem and you can turn it off, you would at least have control, where as blood levels go down, you could decrease the length of the experience. Whereas if you swallow something in a capsule, it could go on for 6 or 8 hours. So those were important. And then the other aspect, when we're talking with the therapist, has to do with the fact that, a patient comes in, they setting, they go in the room, talk with the therapist, they take the capsule. It takes an hour, maybe longer, to go in to achieve the psychedelic state, and then it could last another 6 or 8 hours. So, you could have a patient in a room with the therapist, 2 therapists, for 8 hours or more. And that's a bit of a burden to the therapist and to the patient. And it seemed, and also from, if you're looking at trying to help people and having clinics, that's not very scalable. So the idea with 8803, when I discussed with Robin, is, okay, you don't give an IV bolus, you give an IV infusion over 10 minutes or so to achieve the optimum blood levels. And then you slow down the infusion just to maintain that blood level for as long as it's necessary. It could be an hour, it could be 2. And that gives exquisite control to the therapist to try to achieve the best possible outcome. And it so removes a lot of the variables that are associated with other routes of administration. And yeah, so we have quotes from him, and he's a firm supporter of having the Tryp 8803, the IV Psilocin, as the drug component for drug-assisted psychotherapy.
Yeah, that's so interesting. I've had thoughts of it in regards to, let's say, for example, Comp 360, and how would it be just people like patients showing up to the clinic and essentially like trial and erroring, like from a lower dosage and going up higher, and like how that would work as far as time
No, good
constraints?
question. So good question. So what first of all, you would still have the psychotherapy component, set and setting. All those things are similar, right, Adam? But in the literature, it suggests people would say that around 10 nanograms per mil is a good blood level to achieve in order to have a psychedelic experience. To date, no one's had enough control to really make certain that that's the real level that you should achieve. So we'll be able to do that. We'll do what's called first into healthy volunteer studies, where we've done a lot of PK modeling, where we predict that if you give a certain dose over 10 minutes, it'll achieve a certain blood level, then how you maintain that over a period of time. And we'll do that study, and then we'll correlate that with the MEQ and the different questionnaires. And we also have in our patent filing, we've described a change in EEG patterns that may correlate with that. We still have work to do there. And once we have that data, what we'll do is we'll adjust so that we can give a little bit higher dose, and probably about threefold higher dose, to get to a higher blood level. And so we'll be able to have, say, 10 nanograms, 20 nanograms, 30 nanogram per mil blood levels, and then different durations to hold that there for an hour, 2 hours. In that way, we'll have a small range, but a range of different exposure that we can bring in the clinic. And what I think what Tryp will do is, again, we'll repeat a small study in patients, take a couple doses into those patients so that it's not guessing. We go and we see which of the ones seems to be the best. And once we get that information, then we'll bring that into a larger phase 2 study. But we'll do some fundamental research based upon targets that have been reported in the literature to be responsible for achieving a true psychedelic experience.
Oh, that's so interesting.
Mm.
All right, so to build on this with the next question.
Yeah.
So in a recent interview you mentioned, this is a quote, "If we look at the COMPASS trials and the variability in their results, I don't think it is the drug. I think it is the variability in blood levels." So you mentioned this already. The debate over an oral psilocybin pill versus intravenous psilocin has actually spilled over into various online forums. I don't know if you're reading Twitter, but there was a large discussion on Twitter in regards to this. One prominent voice countered your statement by saying, "It is a big assumption to say it's just blood levels.
Yeah.
What about patient-specific blood pH, alkaline phosphatase activity, polymorphisms in HTR, MAO, and hundreds of other things? Do you have a rebuttal you would like to give to this proposed counter argument?"
Well, I don't whether you call it a rebuttal or maybe perhaps an explanation. So what I was saying is that following oral administration, it's pretty well known that there's variability in blood levels. Okay. And I mentioned a couple things that certainly can contribute to that. And that's known for other drugs. I mean, if you talk to people, different cytochrome P450 in your liver, there's these enzymes, the CYP enzymes. And in drug development, you have to see, what if someone's taking another drug that inhibits those enzymes? Your blood levels will be higher if it stimulates. So people know this, and so it's very well understood. I didn't say that there were other things that could or could not impact this variability, but the variability is there. What I was really trying to say is that people know that if the proper amount of psilocin gets through the blood-brain barrier into the brain, you will have a psychedelic experience. But how do you make certain that blood level is achieved? And we think that with an IV and given the active, not the prodrug, is the best way to do that. Now, I'm also not saying that we know the exact blood levels that would correlate. We have to do that work. But certainly, I don't think anyone will argue that IV isn't more accurate than oral, and that there's first pass metabolism occurs with many, many drugs, and that contributes to variability. And the point I was trying to make when I spoke about that is what I mentioned to you before, Adam, the fact that some of the people who got 0.3 mg per kg had higher blood levels than people who got 0.6, right? So if you look at in general at the Compass 360 study, a patient who got 10 milligrams dose potentially could have had higher blood levels than some of the 25, given just the data that, and I'm not saying this is an absolute, I'm saying it's a potential explanation. And of course, there could be a multitude of other factors that come into play, whether or not people have a clinical response. These populations are incredibly heterogeneous. They could have other things going on. My point is more, no one will argue with me, even on Twitter, that IV is more accurate than oral, and that if you give the active drug itself, not a precursor, that's likely more accurate as well. The other things, we can discuss. I'd be more than happy to discuss with folks. But yeah, we understand that it's more than just a blood level. You look at, we're looking at fibromyalgia. We're looking at eating. That's a very heterogeneous population. I'm not so bold to say, oh, we're going to get the right blood level and we're going to cure everybody. That's not the point. We're just saying that if the drug component is important, let's make sure that at least the patient gets the requisite exposure that we think is necessary. Does that make sense? I think hopefully that diffuses some of that that discussion.
No, it definitely makes sense, and also I can say, so in my work that I've done with psychedelics as well, it's very well known that there is no set dose that you can give a person in regards to oral, like let's say shrooms or magic mushrooms, that it translates across the board. It's variable for every person, and I think that in what you're to build on what you're saying, it's definitely intravenous would definitely be a more efficient way to kind of figuring out what that dose would be for a specific person.
Adam, think about even other areas. A doctor will come and say, okay, start with this dose, and if it doesn't work, they'll raise the dose. And you mentioned that before. You could start out with a lower dose in a particular patient population, and if they don't achieve the benefit, the clinician could decide, hey, you know what? We'll give a little bit higher dose. With the IV, at least you're getting a higher dose. And that's the point. Mm-hmm. And I think there's still work to be done to really generate the data on how you correlate blood levels with the psychedelic experience with a positive clinical outcome.
Understood. And so this is something that just popped into my head, actually. So usually when people, so I have only experience with oral psychedelics. If somebody takes a dose and then let's say they wait 45 minutes and they take another dose, you have this inbuilt tolerance that kind of starts to build up very, very quickly. Is IV able to kind of circumvent that tolerance?
Yeah. So in a way, so what you're doing is, and it's actually a really, really interesting question. So if you look at most of the studies that are being performed now, you'll have an intervention, right? And then 3 weeks later, you'll have another intervention. And given our discussions with Robin and other folks in the field, they like that idea because they think that you could have very profound experience with the first one. And then the second one can reinforce that and help the durability of the response so that the benefit extends out further. In pharmacology, there is a thing called tolerance, where if you have too high drug too often, the number of the receptors on the surface actually decrease. And so you would develop some level of tolerance. What we're looking at here is, so let me take a step back because I may not be explaining this. If you swallow something orally, your blood levels look more like Mount Everest. They're peaked, they go up and they come down. What we'll do with the infusion is going to be a plateau. It's going to go up, not nearly as high, and stay here at lower levels. If you look at the side effects like cardiovascular or some of the others, they're associated with that peak level, not a lower level that's clinically effective. So our total exposure could actually be a little bit lower because we're not going to have that big peak. One of the things they call area under the curve, which is that whole area might be as large or larger, but our blood levels will most likely be a little bit below those that you traditionally get with oral. So I think that the likelihood of us generating tolerability is low. And the other thing that some of the therapists have said to us is they said, Jim, what we're thinking with the IV is could be that, okay, 3 months out, 4 months out, they're starting to experience some of the problems. They could come in and have a very short experience. It could be a half hour, just come in with the therapist, sort of reinforce maintenance dose and do that. And we're certainly thinking of other routes of administration that could be used for the maintenance dose. But right now, we just want to show a benefit, right? And then we can think of creative ways to maintain that benefit.
Oh, that's amazing. So like efficiency seems to be the word here, where like it keeps popping into my brain, where like it just everything that you're working on sounds just very efficient.
Yeah, Adam. So think about this. So we work with Fluencent, and they're really, really good. They do our psychotherapy training, and I was speaking with Ingmar, one of the founders, and he was saying, Jim, what's sort of neat about this is if we know you're going to have people going in with say 15 minutes or whatever, and it's predictable, and you have control. He goes, I was just thinking you could have sort of like a central monitoring system and just one therapist per room, and so it'd be less of a burden. You wouldn't have to have as many therapists. You could have more sessions being performed. So there's other things I hadn't even thought of that might be a benefit when you with the IV, because it's all controlled, right? You sort of know when it's going to happen, how long it's going to happen, and you can be better prepared.
Amazing. Okay, let's get to the next question.
Yeah, sure.
So, given the importance of set and setting, how do you address concerns over needles and the potential constraints around IV administration with TRP 8803? Have you considered eventually utilizing some sort of subcutaneous pump device to administer 8803? Last part. Also, can you please speak to the IP protection you have around 8803?
All right, boy, you were loading them up there, Adam. Yeah, so we've had that discussion. It was interesting. We talked with the people who are interested in, and the one thing was that I found interesting where people were anxious about the idea of swallowing a capsule and losing control. So they like the idea of having that control element with the IV. And we talk around, we talk to other folks, and the idea where you're going to be in a couch, you have blindfolds on, music, and you're not doing jumping jacks or running around the room. You're trying to look inwardly, and there are things that you can do with swivels and things where it shouldn't be cumbersome. Could you have the issue with an implant though is that you would have to predetermine ahead of time exactly what dose and how long. I'm just thinking it's a great idea, but the idea with an infusion pump is you have total variability, total control where you can change things. So I'm not saying there isn't a better mousetrap, but right now, I think what we'll be able to do with 8803, we'll be sort of one of the first ones to be able to start figuring out what are the ideal blood levels, what's the ideal duration of the optimum duration, because we'll have that control. After that, there could be more innovative ways than having an IV. I mean, you could have a mini pump that you just tape to the arm that has a set flow. There's certain things that I'm sure you could do. I haven't given that too much thought. We're just trying to get through the first part. With respect to IP on 8803, it's a very interesting question because there's certain corollary to it. One is that, yeah, so it's a different molecule, different dose, different route of administration. It's an infusion over time, so it has a lot of bells and whistles. We have the EEG data there. One of the things I can mention, because we published this now, is we did a study at the University of Michigan in a rodent model of fibromyalgia, and we gave a single IV infusion over 60 minutes, and it reduced pain in that model for 4 weeks, 28 days to the end of the study. So it's an animal model, but it shows that this infusion approach, at least in the animal model, works. So when you have data like that, that helps with your IP. And I'll come back to IP with another comment. What also happened, because this was seen, the patent lawyers were really excited about this. It's totally, we're not giving psilocybin. It's not oral. It's not a polymorph. It's not a salt. And we have salts. We have all that for our psilocin as well. But what happened with that is FDA also told us that it's novel, it's different. And so we had to perform about 7 different bridging toxicology studies. So we spent a year and probably upwards of $1 million just to do all the qualifying studies with psilocin in order to be able to go into human studies. So not only is it seen unique from the patent perspective, but FDA also saw that we had to do some additional work. The other thing in my background, as I said, in biotech, is at the advent of biotech, if you were cloning, you were creating in bacteria insulin or other normal, you can't patent that because those things exist in nature. So it's very difficult to patent natural products like psilocybin and things. But what we started doing is, since we were looking at different areas, right? So the point being, let's take binge eating as an example. So we filed a patent on the use of psilocybin and its active metabolites for the treatment of binge eating. And you file a provisional patent, and we put our protocol, the endpoints of our protocol in there as claims. So now we've just announced from our binge eating study that we saw an 80% reduction in binge eating episodes, 84% reduction in the patient's sense that they lost control over their eating behavior. We saw improvement in depression, improvement in anxiety. So now we have data to support that patent. It's pretty slick. So we're doing that with fibromyalgia, we're doing that with IBS, so that we have multiple layers of patent protection, not solely around 8803, although we have that as well. So that I call it the pick a fence approach. So we have, we got a fence, we got a moat, we got, a river, and we have all these different things to protect the areas that we'll be working in.
Wow, that's remarkable. And in terms of the results that you're getting, let's say specifically for eating disorders or a binge eating disorder, that's phenomenal. That really is.
Well, yeah, Adam, everyone who knows, I'm like the world's biggest optimist. And so we knew we do these open label studies where no placebo, everyone gets the drug. It makes enrolling a lot easier. People know we're going to try to help them. And so Dr. Miller and Jesse Dowry, the lead psychologist, afterwards would just talk about how pretty remarkable, the changes were. And they really, without having all the data, they really felt that there was a tremendous benefit to the patients. And so it's a little bit different. When you do a double-blind study, they call break the blind. So you see all the results, but you don't know who's placebo or who gets drug. Excuse me. Here, we knew everyone got the drug, but we didn't have the final numbers. And to see that 80% reduction, that effect size is pretty remarkable. And to see the benefit in other areas is really rewarding for the patients, because here's one of the things that made me change my mind. Again, having been in this field for a long time, sometimes, this sounds horrible, but you look at a patient as an indication, not a person. Oh, this person just has pain, but you don't realize that this person can't sleep, or they're depressed about it, or they're anxious about it. And I think the approach that we're taking is looking a little bit more holistically at the patient, not just that singular endpoint. And it's a totally different approach. And we're, unlike a lot of drugs, like if you in the pain space, we're in fibromyalgia, about a third of those patients resort to opioid use, which obviously is a problem in this country. But you're treating the symptom, you're not treating the cause of the pain. Think about, we were talking about phantom limb. You're treating that symptom, not really what's causing it. And here we have the ability to go a little bit further up and alleviate what could be what's causing the actual pain. So it's a really different approach than what's standard, in the pharma industry.
Absolutely. So let's get to the next question.
Yeah.
So, and I just want to say also, before I get into this question, so I hadn't even considered the fact that using IV administration for psilocybin, because you're so focused on the needle. But what you said in terms of having control, I think that that's extremely valid, and it really blew me away when you said that, because a lot of times when I've worked with people in terms of with psilocybin and whatnot, that's the number one thing that people are so scared of. It's like they want to have control, they want to be able to end it if they're not having a good time. And I think that you're addressing something that so it can really put people at ease and make them
and think about it. You have a blanket. It's not like you're gonna be sitting there like, oh my God, I have a needle in my vein. That's we're hoping that we can establish a little bit different environment. But I was surprised too. As I said, there was a couple people and they go, Jim, but I really want to do this, but I was afraid of this whole thing about the lack of control and having just better accuracy. From my standpoint, I came in as a chief scientific officer. Now I'm CEO, but you want to give the drug the best chance of success, right? And that's one of the biggest problems in drug development, that people don't select the right dose, and they're underdosed or overdosed. And so let's give the molecule the best shot. And by giving it IV and getting to that level that we think where we expect it to work, at least we know. And I always tell stories. It's just my background. So the board was asking me, so Jim, how do you know if it's going to work in binge eating or not? And so I can't be serious. I'm sitting there, well, guys, I said, if the first couple of patients, they get the munchies and they're eating more than before, it's probably not working. If on the other hand, we get a reduction in their binge eating episodes, then it seems like it works. And obviously that's what happened. So, I think being open-minded going into these things is the most important thing, and not feel like you know all the answers or whatever. You have an hypothesis, and until you get the data, it's still just a hypothesis. So,
thank you so much. That was a really solid answer. So, all right. So to build on that, is using TRP 8803, how quickly can the trip realistically be shut off? One critic has mentioned, outside of ketanserin administration, which has its own issues, I don't think it can be turned off as quickly as you think. The half-life of psilocin is 1 to 3 hours. So I'd love to hear your response to this.
Sure. So now I'm going to go into a little pharmacology. I'll use my PhD in pharmacology. So one of the things that people have reported in the literature, again, not me, is that you have to have a certain number of receptors, the 5-HT2A receptors in the brain, have to have the drug on them in order to have the experience. Okay. And so say that at 10 nanograms per mil of the blood, you have the number 65% in the literature, whatever it is, but it's a number. If you're right on that border at 10, okay, and you stop, of course, it's not immediate, but as the blood levels come down, receptor level goes down and they start coming out. They're absolutely right. If in this patient we decide that they needed higher, that they were 30 nanograms, okay, then a half-life would be 15. So they would be in the psychedelic state longer. So it does depend upon what your target is, but certainly being able to turn it off is much better than if you had an implant, that's going to degrade, or oral where you have no control. So I'm not saying it all I'm saying is it's better. And it depends upon where you are. The other thing, I'm not going to go into too much detail. We've done a fair amount of PK on IV psilocybin and IV psilocin. And our results on half-life are a little bit different than what's seen when it's administered orally. And one of the things with oral is you have what's called an absorption phase where, as it's going down, it's continue to come in, right? And so what you get is a balance between how much is coming in and how much is going out. And that's how you get. Whereas if there's a paper actually from Robin where he gave a 1.5 mg IV bolus, and I think the duration, I could be wrong on this, so people get me on Twitter, but I think the duration of the psychedelic experience was like 15 minutes. So pretty clearly with IV, it comes down pretty quickly. And I'll have my position, but I can't argue that until we actually do. The other thing is, if you look in our patent, looking at EEG, when we stopped the infusion, the change in EEG was, let's just say, more rapid than the half-life that's been reported following oral administration.
Understood. And anyway, even though, just because so without even thinking about, let's say someone's having a bad experience, even that they're under IV, wouldn't it be easier to give something if like an extreme situation, like that would antagonize the 5-HT2A
you give it
receptor?
serotonin like canacerin, and again, you're not going to do that routinely, but if it's something that's really serious, it does afford you the opportunity to do that. And again, we're not saying this is going to be common practice. It's like, all right, time's up, we're going to give you a serotonin antagonist. No, it's in a medical emergency, something that you could do. Yeah, so you get it.
Yeah.
I'm going to have you argue with these guys on Twitter for me, Adam. I'm not going to bother.
Yeah. All right, so up next, what we have here is, as of November 30th, 2022, Tryp had about $800,000 in cash.
Yeah.
How much cash does Tryp need to fund its operations for the full year, and how soon will the company need to raise more capital? How confident are you the company will be able to successfully raise new capital in this current market environment?
So wait, Adam, you trying to tell me it's a tough market to raise money now?
I mean, that's the word on the street these days.
Yeah, I don't think I had any gray hair before we went into this. So one of the things that we have a benefit is our major investor, EGB Ventures, and Bill Gardner is a solid supporter, and it's nice to have that as a backdrop. As all biotech companies, we're exploring opportunities for investment. And the interesting thing with our model, we're a virtual company. The amount we pay our there's only 2 employees is low, and then everyone else is on consulting. So our burn rate is quite low. Some of the relationships we have with the universities help us do things very cost effectively. So, for example, God bless MGH, they have some grant money, so that the cost of doing that study was deferred by some of the grant money, the money that they had at their disposal. So we're able to do things fairly cost effectively. And it's no secret, we're at JP Morgan. We're looking at any number of ways to raise capital because we're planning for success. We want to take 8803 into patient studies. So right now, excuse me, we have the fibromyalgia study will be starting pretty quickly. One of the questions I usually get is, what's the biggest hurdle, in doing your clinical studies? And it's actually the DEA. It's getting DEA clearance on a state-by-state basis for the different sites to be able to start the study. So fibromyalgia will be starting, and folks at University of Michigan are just phenomenal. They're the best. And then we'll have IBS. So this year, we'll be having that data come out. And we will, as I think I mentioned, we're looking at potentially Australia, UK, Canada, to do these first-in-man studies. We can do them a little more quickly there, more cost-effectively. And we're exploring every opportunity we can for I would say metered or financing, where given where the markets are now, where our valuation is, to take in the requisite funds we need to get to key inflection points, which we can discuss, which I alluded to, and as those things come out, avail ourselves to additional capital. And we're looking at that. We're not adverse to what could be a strategic partnership. In the past, I went to the dark side back in 2000. I got an MBA in international business, and so I've done transactions. I've done deals with GSK and Novartis and big pharma. So, there could be opportunities where we look at licensing for a global area. It could be Europe or Canada or Australia. So that we're very open-minded. We're painfully aware that the investment in healthcare biotech is down, but my sense is the overall sector is coming up. You have Compass has good results. You have the MDMA results. You have different. We've had our binge eating. We expect to have good data in fibromyalgia and other areas. So we think it'll become more attractive to the investment community, and we're out talking to everyone. So we started this off where we had a low profile, where Peter Malloy, who just came on in December as the chief business officer, we're out speaking to everyone every day. Some days I can speak 8 to 10 different people. So we're hoping to get our visibility higher, for people to understand the Tryp story, to be part of the conversation. And we, like all biotech companies, clinical research is relatively expensive, and so we'll be looking to raise funds. We're speaking with folks now, and again, we're fortunate to have the support of EGB so that we can do things modestly to meet our demands going forward. We brought in Jim O'Neill as our CFO, so we have pretty tight budgets, and so we're doing things cautiously. But we think we're in pretty good shape.
Nice to hear. So to build on that a little bit, or actually, this is kind of like a side tangent to that, but relevant still.
Sure.
So can you give a ballpark of how much the 8802 binge eating disorder 2A trial cost?
I don't. So let's just say a trial in this sector, from our experience. And this is across different indications. It's not just the clinical sites. You have what's called your CRO, you have your data management, you have monitoring. So to do GCPs, I would estimate a small study, a 10 patient study, between $1 and $1.5 million would be a reasonable, because you have the psychiatrists, you have the training, you have all the therapists. So it's more expensive than some of the studies I've done in the past. So I did studies in osteoporosis where it was 1 year treatment, BMD. And in those studies, you would say, oh, your cost is going to be $30,000 per patient. Because it's a little bit different. You give them a bottle of pills, go home, take them, and come back. And, here it's much more intensive. We're doing fMRI, we're doing neuroimaging, we're doing a lot of things that make these studies, a little bit more expensive. But yeah, I think that's a fair estimate of what the cost is for these types of studies.
Thank you for being so candid about that.
Yeah.
So to build more on William Gardner. So co-founder William Gardner owns about 40% of Tryp's outstanding shares and has contributed a meaningful amount of capital to Tryp thus far. What is Gardner's current relationship with the company, and is he willing or able to further invest in the business if need be? If you can answer that the best of
so he's certainly able, from every indication with weekly calls. I think he's willing. Again, look, it's in the public domain. Last year we had a change in management, and so we've moved on from that. And I think what we had to do was buckle down and focus on our key areas in advanced Tryp 8803. We've done that, and EGB is very happy with the results to date. We EGB host a dinner right before JPM, and we were there. We presented, and the information we had was well received. So I think we're doing a good job, and we have EGB support, and I think that that's important, because you don't want to be in a position where you have to do a deal that ends up in the long run not being the best for the initial investors. So we're trying to protect the investors, not just EGB and Bill, but other people have spent their money and got invested. And hey, look, we're all disappointed. Everyone was in the same. You're in the field. We went from 15 cents to $1.15 and down to 15 cents, and that happened to Simon and Compass, and everybody's been on that roller coaster. And what the best thing I can do as a CEO is build a sound foundation, right? So we get a good foundation that we can then expand upon. And I think with 8803 and our core indications, we have that. We have interest in other indications, but with the current resources, we have to focus on things that are at hand. But that's part of the reason why we speak with many different groups to see, hey, do you want to co-develop something? Could we work together to advance a program? Is there something, creative we could do along those lines? Is there something we could do with groups outside the U.S. To do things? So we're very open-minded and willing to entertain anything that we think is good for the company and investors. And that's my story, and I'm sticking with it. So
no, that's very good to hear, and especially in terms of the way you're speaking about taking investors into account, because I think a lot of times when people are invested in some of these companies, the retail investors kind of feel overlooked a lot. So it was nice to hear you say that.
Oh, absolutely. They're super important, and I personally, we have some investors who own, say, 1 million shares, whatever. If they want to get on the phone, I'm not going to give them any insider information, but I'll explain to them what we're doing. I was executive vice president of Unigene, which was a biotech company, and one of the things I did routinely was talk to different groups, and so that they understood what we were doing. And the more they understood, the more they appreciated our strategy. And I'm doing the same thing with Tryp, taking time to speak to people and say, here's why we're doing these things, and here's the steps. I'm giving away my secret sauce now. What you're telling us, here's what's important for success. So when you achieve those, they recognize it, and you get more enthusiasm, and you want to build a good investor base. You want people to be excited about it and understand why you're doing things, and that when you do achieve them, how important it is. And speaking to other people, just to get the word out that, when, it's funny, Adam, people say, look at the sector, right? And so, I play sports, I played basketball in college and all these things. So I do different analogies. But here I'm saying, if you want to get involved in this sector, which I think has tremendous upside potential, you want to pick a horse, right? Who's going to win? And we think where Tryp is right now and what we'll be achieving in 2023, we're going to be a good horse to bet on. I think every CEO says this, but we're all incredibly undervalued right now. So it's a great time for investors to get involved, because not going to go much lower. And if you want to get your 8x, 10x return, now's a good time. Yeah, there's risk, but there's tremendous upside in the sector in general. And certainly, at Tryp, we believe that there's tremendous upside.
Agreed. I agree with you 100%.
Yeah.
So we're winding it down here. So the last question we have is, where do you think the psychedelic industry is at right now, and where does Tryp fit into this picture? I mean, you kind of just answered that, but what are your opinions on industry consolidation, M&A, availability of capital, and overall investor interest in the sector? And then to tack on just one thing at the end there, you mentioned at the beginning, we mentioned near-term catalysts that investors have to look forward to. If you can kind of Elaborate.
I'll tell you one thing's interesting. The TGA in Australia, you probably saw that. As of July, their psychiatrists there, certain appointed psychiatrists will start being able to administer to patients MDMA for PTSD and psilocybin for TRD. So that's a monumental leap where you have a regulatory body, almost like we had the EUA here during COVID, emergency use authorization. So I certainly see things changing. You look at the quality of the universities doing psychedelic research now. You have Harvard, you have Yale, you have Hopkins. I mean, across the board, leading institutions. You have NIH funding grants. So I think the stigma from, well, you're too young, from the 70s, but I remember on psychedelics is dissipating. People are freely discussing their use of psychedelics, that they've had a beneficial effect. You just see data starting to come out to support this. So we've made a contribution with binge eating. We expect to make more contributions, and I'm a firm proponent that rising tide rises all ships. And I think the sector, and look, you're going to have more data coming out. There's a high likelihood MDMA is going to be approved for PTSD. I think I do believe Compass will have positive data. I think we're going to have positive data in fibromyalgia and IBS. And so the more rigorous science, the more credibility you get. And, I say this to folks. So again, this is self-serving, but from a Tryp perspective. So we get benefit in fibromyalgia, phantom limb, and maybe complex regional pain syndrome. We have a pain franchise. We would become a very attractive target for a big pharma who wants to get into the pain sector that's non-opioid, right? That's unique, different mechanisms of action. So you can see where there's tremendous upside in the fields. And with the more, as I said, I'm repeating myself, but with the more groups doing clinical research with positive data, look at NYU with alcohol use disorder, 80% reduction in alcohol consumption. I mean, these are big numbers, and I think the more people see this, I think there'll be a revitalization in the sector. Part of what could have happened, again, I'm the CSO and I'm the science guy, but I think people saw what happened in the cannabis sector and they just thought the same thing was going to happen in psychedelic, and it was going to be immediate, but it takes time. But I think all in all, there is positive momentum in the sector. And I think people will come back to it, both on the retail side and on the investor side. And I think pharma is going to start taking notice as we progress as well.
Agreed. The excitement about the psychedelic sector is only growing, even with the market conditions that we've been experiencing. The excitement is still there. The excitement is still there.
Exactly. Yeah.
So thank you so much for sitting down to have this interview. I really, really appreciated talking to you, Jim. And I hope to have you on again at some point soon, and we could kind of touch base.
Absolutely. Anything comes up, Adam, I obviously love talking about this stuff, so it's not a problem. All righty.
All right. Thank you so much.
Take care.
I hope that you found today's interview to be informative and enjoyable. If you'd like to see more content like this, please take a moment to like and subscribe to the channel. My commitment is to bring you high quality and engaging discussions, and there's certainly much more to come. Thank you for the support, and I look forward to your continued engagement. If you'd like to reach out to me, if you have any interest in being on the show, I'll have all my contact information in the description of this video. Thank you so much, and have a wonderful, wonderful day.